Okay, this is just to finish the biomolecules. We're on our last biomolecule of the 4 nucleic acids. The monomer is called the nucleotide, made of a phosphate group, a sugar, in this case it's a pentagon sugar, a base, a nitrogenous base. This is the simplified diagram that you can use. Remember that this is the part that changes. The nitrogenous base changes from one nucleotide to another. So for example, here, the sugars and the phosphate groups are the same, and the bases are changing, okay? 4 of these are used for DNA, 4 of these are used for RNA. How do these nucleotides work? So here are 4 nucleotides. That's one, that's another one, that's another one, that's another one, right? Each nucleotide has phosphate group, sugar, nitrogenous base. And look what happens to them. They get attached here. One sugar attached to a phosphate group, sugar, phosphate group, sugar, phosphate group, and so on. So they attach to form the polymer. So we have monomer, nucleotide, polymer, nucleic acid, either DNA or RNA. A nucleic acid is the polymer, a whole string of nucleotides attached together. What's really important is that when you attach them, notice what you get. It looks, for DNA, it looks a little bit like a ladder. There's the legs of the ladder, and there's the rungs of the ladder, the horizontal parts. The legs of the ladder are phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And remember, these are unchanging. Same thing on this side, phosphate sugar, phosphate sugar. In the middle, we have the nitrogenous bases, and the bases are distinct, and they also match up with their corresponding pair. So here they show it by shapes, and that's all we need to learn is, since we're not understanding the real chemical formulas of the nitrogenous bases, the shapes are a perfect indication for us to understand that these match up as pairs. So cytosine and guanine match up, and when there's guanine on one side, there'll be cytosine on the other, and vice versa, and same thing with other sets, okay? And so there's this base pairing, it's called, that happens between one strand of nucleotides and another strand of nucleotides, and that's what creates the double-stranded DNA. Here's the same exact thing, but more chemically accurate. This is more chemically detailed, right? Phosphate sugar leg, base pairing is happening in the center. And the base pairing is precise. And the base pairing is very important because it carries the information. This code for the genetic code and the genetic information is within this code of the order of the base pairs. And so that's why that's super important. This is the same thing, just more chemically detailed. And just to remind you, the RNA is single-stranded, the DNA is double-stranded, and they both kind of coil into a helix. Here they're just showing you the types of nitrogenous bases. Each color represents a different nitrogenous base. Here is a summary of the 4 biomolecules. So remember that pentagons and hexagons are always sugars. These are meant to represent hydrocarbon chains. Each one of these is not a circle, but it's an amino acid. And this, of course, you should recognize the nucleotide. So hopefully you can recognize what each one of these is, right? Nucleic acid, actually this is specifically RNA because it's single-stranded. And here's the monomer, nucleotide. Carbohydrate or polysaccharide is the polymer, and here's the monomer, monosaccharide or sugar. Here is the polymer. It's a lipid, specifically it's a triglyceride. And what are the monomers? They are fatty acids as well as glycerol. Protein is the polymer, specifically a polypeptide chain, and the monomer is amino acid. So this is a really good, this is a really good review slide. You should notice that there's one thing missing here. I hope you noticed it. There's 2 other types of lipids. So if this diagram were more complete, they would add the phospholipid, the little jellyfish molecule, the phosphate with the 2 fatty acids, and lastly the steroid, even though the steroid is kind of odd. I think that is everything we need to know. Yeah, I think we're all done.